We have a classroom still to come in, so I'll talk a little bit and introduce our seminar speaker for today, who is Julio Harvey from Mbari down the way. Julio, I'm very happy to shake hands and meet today because we've had several interesting conversations by telephone, but honestly haven't had, so this will be very exciting for myself. Our lab group knows about your work, and you're meeting many of the BioOcean students and many in the room here. Julio is semi-local. He went to school with Diana Steller in graduate school. He graduated from earlier undergrad degree from Cal Berkeley and did his PhD up at UC Santa Cruz with Linda Goff and spent a few years up in the Puget Sound area in a technical position, dabbling in the area of identifying, monitoring, and developing methods for aquatic invasive species, and spent a little bit of time searching for a job, and now has been for several years. I tried to memorize 5, but it could be 7, but it's probably 5 years working in Bob Breinhoek's laboratory, working on molecular techniques in invertebrates. He's actually a jack of all trades with organisms and is perfectly willing to look at, tear apart, or sequence nucleotides and just about anything. He's worked professionally on fungi, terrestrial, on phytoplankton, marine, probably in some fresh water. He's worked on seaweeds themselves for his thesis per se. He's worked on invertebrates, which is his current avenue of research, both juvenile stages and adults, which is a good thing to be working on. Don't forget that there's more than one look to the organism. And he's also worked on sequencing mammals, but that's only because of sloppy lab technique, and he managed to sequence himself a couple times in the laboratory, but he doesn't really talk about that. And he's currently working on exciting projects dealing with molecular techniques applied to sampling, all of them Mbari way. And so, no further ado, we'll give an warm welcome to Julio Harvey and his talk on his book, Current Research.
Thank you very much for that wonderful introduction. It's also a pleasure to meet you as well. And I hope that my time in school with Diana is seen as something that will give me oodles of street cred and be a feather in my cap rather than a black eye. But you don't know her like I know her, I'm pretty sure. And so I just don't know. I'm going to have to wait to find that out. So thanks for inviting me over across the bridge today. It's kind of ridiculous, but I realized that the last time I was in this building was about 5 years ago, despite being a quick walk away. So it's great to come back and be able to talk to you guys about some of the stuff that we're doing. And the title of the talk today, we're talking about adaptive sampling of these robots. Basically, the main reason I came over is not so much for the robots, but for some of the work that's going on in Nick's lab. Looking at ballast water and talking about detecting various kinds of things in ballast water. And this methodology is something that I'll focus on a bit in the talk. But the sort of point of this intro slide is that we do have a situation that many of you are familiar with, where in coastal oceans there are a lot of physical forces operating and a lot of physical processes that zooplankton and phytoplankton have evolved to take advantage of. And because these processes are so temporally and spatially variable, it's very difficult to actually directly sample features like upwelling fronts, thin layers of chlorophyll and phytoplankton blooms, intermediate nepheloid layers, and other features, unless you have $1 million robot to do so. But I want to make the point up front that although I'll be talking quite a bit about robots, this chemistry and this methodology is very accessible economically. It's something that any lab with moderate funding should be able to afford and put to use. So I'll just start by preaching to the choir a little bit about some major questions. How are plankton transported? I already mentioned the important or the mechanisms behind that, but that's important because of dispersal issues, particularly involving marine protected area reserve designs and informing fisheries management. And due to before or after transport, what things happen with aggregation? Why is that important? Well, it's important for ecosystem structure and function. how everything works out there in terms of fueling the ecosystem economy, if you will. And then also the idea of local retention, as you may probably know. In recent years, this has been found in many cases, especially for broadcast flying invertebrates, to be the rule rather than the exception. And mechanisms for shoreline delivery and recruitment are also important. So one of the key questions, though, for stuff we're talking about today is what physical mechanisms mediate these processes? How can we identify those with these vehicles, and how has the biology evolved in the use of them? So for just a brief overview, there's sort of three parts of this talk today. The first is I'll tell you about the robotic instruments involved. And second, I'll spend probably a pretty great deal of time talking about the molecular method, because I do feel that this method is something that might be applicable to ballast water questions. I actually worked with ballast water myself for a bit at Seattle, as I mentioned, and ever since I tried to do that, I've been looking for a way to do it right, because we weren't exactly, in my opinion, doing very well. And then I'll finish by giving some examples from studies of plankton ecology with AUVs, among other methods. So I just want to briefly mention that the AUVs and the programs we have in MBARI are part of this overall ocean observing system that I'm sure you've been involved with or heard about. And it spans everything from remote sensing to local moorings, standard oceanographic work from ships, Minskin rosettes, nets, and of course the robots as well. We have different kinds of AUVs available to use. We have these long-range AUVs developed by Jim Bellingham's lab, and they don't water sample. The AUV Dorado class vehicle is the one we talked about the most today, and they do sample water in addition to a variety of other things. And then we have stationary robots like the ESP that many of you probably know have developed over the past decade or so by Chris Shulman's group, and currently in its third or possibly soon fourth generation. And along with the ocean observing system comes a myriad of data sets that are very interdisciplinary and incredibly hard to look at and synthesize all in one place. And so the other aspect of development not pictured here is that we have software engineers working on systems to collect all this data in one interface so that we can analyze it, look at it, and Mike McCann's been doing a lot of that work for our projects. There's a spatial temporal oceanographic query system, cutely acronym STOKES, that we now have that is invaluable in my work, and it's basically taken sample analysis of sample data from maybe a 3 or 4 week prospect and compressed it probably by 2/3. So it's really key to note that these databases are going to become more common. And the other thing, like a lot of the stuff that we do, it's all open access software. So if you have any designs on trying to do something like that, you can contact Mike or contact me, and we can put you in touch with a lot of the scripts and stuff that we've done, and literally use it for any kind of data. So I'll start by just briefly mentioning the environmental sample processor that as I mentioned, you know. It's a lab in a can, stationary, hangs on a mooring, or it can actually be, we've got a titanium sphere we can put it in that's rated to, I think, around 4,000 meters. There's also a drifter application that they use, I think, up near Station of Loha, Blackline Islands. And essentially what this device does is it slowly sips in water, and then it does all the lab analyses that you need to do on shore in situ, and reports back the data in real time. And so it was originally developed by Shulman's group to monitor harmful algal blooms, and it's this same detection chemistry that was the original chemistry for this machine. Now they use several other methods as well, including a qPCR module, but the original detection methodology is something that in our lab I modified in order to use with cell pumping. And so now what I'm doing is I'm using that same chemistry to work on the water samples from the AUV. And so just to introduce the AUV, the Dorado AUV gulper, and it's the gulper because it's got these 10 water sampling syringes on it. Probably in the next 4 months or so, we're going to increase that number to 20. And you've got these ports on the side of the vehicle. And so what the vehicle can do is it's cruising through the ocean, taking environmental data as it goes, and based on the environmental data streams, it can trigger water sampling, or you can set predetermined waypoints. But one thing to note about this vehicle is typically it profiles in the water column, so it swims these up and down goes, and you'll be able to see some synoptic hydrographic data of that in a little bit. But basically, what you've got is a situation where you have these water samples, and you've got anywhere from 12, maybe 8 to 12, or even I think it can go up to about 16 hours deployment. And you've got a vehicle that's also collecting, CTD data. It's got redundant CTDs. It's all got a chlorophyll oxygen sensor, has isis nitrate developed by Ken Johnson's lab in Bari. It's got several Sequoia products on it, laser in situ devices, and it also has a list holo on it that Heather Fulton Bennett was an intern last summer worked on. So a lot of bells and whistles, a lot of environmental data, and the water samples to go with it. And so what I'm going to do here in this first section is tell you about 2 out of the, I think there's 4 or 5 that have been developed total, adaptive sampling algorithms that have been developed in Jim Bellingham's lab by Young Jong, who's a software engineer that came to us from MIT a number of years before I got there, and he's making all our lives better. He's a wonderful guy. What can I say? He's developed these elegant capture programs. This algorithm, if you look at this cartoon, what it does, you've got a chlorophyll thin layer here depicted by this red. And the problem we used to have was we figured out we could tell the AUV to detect when it detected chlorophyll to fire the voltors. But as you know, a lot of these layers are really thin, sometimes as little as a meter or less. And so what would happen is the AUV would just pass right through the layer. And it would be on the other side by the time half a second it takes for the algorithm to tell it to fire. So what Young came up with was a way to have the vehicle actually save a sliding boxcar of local environmental signals, and then the vehicle could record how long it would actually take it, what the roughly 1 second delay was from the detection. And so what would then happen would be it would go up maybe past the chlorophyll layer, and then on the downward, or it could be upward, either way, the peak would be anticipated, and the vehicle is able to preemptively tell itself to fire, and the water sample is collected spot on. And it's amazing how well this works. It's actually published here in this robotics journal, engineering journal. And let's look at an example of how well it works. And one thing I'd like to point out about this is, first of all, we've got chlorophyll fluorescence on the y-axis and elapsed time, which can also be seen as AUV distance on the bottom. And you can see these black triangles where the samples were taken are at chlorophyll peaks, but they're local peaks, right? So you got very low chlorophyll here, but that was the highest peak for the location. So it really works most, if not all the time. I like to say 9 times out of 10. And here it seems like it's a bit lower, but these are big, clunky triangles. And really probably what's happened here is that there's a peak about that height here, and then the next one is higher, but it's beyond what the vehicle had seen. So incredibly excellent algorithm. And then a few years later, we developed a method to incorporate this into upwelling front sampling. So the way that this upwelling front sampling algorithm works is that you have that sliding boxcar that I mentioned for the environmental data, and so that's sort of the average of previous. The thing that's important here is the vertical temperature change, right? So you start with that, and then you set a threshold based on remote sensing, or sea surface temperature, whatever data you have available, to tell it how much temperature change do you want to detect. And then if your now temperature vertical is detected, then it allows the vehicle to start collecting. In the front, just within the front, is the AUV profile here. These arrows correspond to different profiles. Now, another way to look at this is maybe a bit easier to understand is that you've got really 3 water types here after, hence the name of the algorithm, 3 waters. You've got the stratified water column, in which delta T vertical is very large. You've got the upwelling core of the upwelling filament water, the delta T is very small, it's all really cold. And then in the front, the delta T changes quite a bit in the horizontal. And so that's the thing that the vehicle takes advantage of to identify the 3 different water types. And when we send it out on a kind of linear transect, and this is, as I mentioned before, you've got your up and down vertical profiling. So this is temperature data. And if you have a near shore on this end of the transect and offshore on the end closer to me, the AVs coming along the circles that represent stratified samples, the diamonds front samples, and the squares in the upwelling core, the upwelling filament. You can see that the vehicle indeed does as it's programmed to. And one key element here is that for these first two here, the stratified front samples, it's also using the chlorophyll peak capture algorithm simultaneously. So capture chlorophyll peaks out here, and then just go out into the upload core. And you see this is sort of a negative control a lot of the time because very little has had a chance to develop. So we'll shift gears a little bit here and talk about what we actually do with the samples. Here's the gulper here, and we're taking that water and filtering it. I just want to take a moment to thank people like Lisa Ziccarelli here, and Heather Fulton Bennett, and Chris Walls, Shannon Johnson, and Mariah Salisbury and Barry, who've done a great deal of helping me out on the Carson. And we used to actually bring this water back when we had our other boat and do it in the lab, and now we're actually doing it all on the Carson. We're also doing a lot of other traditional sampling that I'll briefly mention later. But what we do is we have this 25 millimeter filter format, and we send a couple of liters through a few filters, and we have three sort of different ways that we go with it. In one case, we use RNA layer to preserve, and it turns out that's actually quite good for microscopy, far better than ethanol in my opinion. I don't know, I think that the jury's probably still out on the formalin, but I don't like to use formalin, so for me it's great. And then we can individually sequence individuals that way. 95% ethanol used for community stuff, and also next generation sequencing. We also put our toe nets in ethanol, unless we're sending them to one of our collaborators that uses formalin. But then the technique I'm going to talk to most about today, the sandwich hybridization assay. The best ways to store the filters for that are either the liquid nitrogen method, or you can also use RNA later. The liquid nitrogen tends to be the easier way for us. And so the sandwich hybridization assay is a two-probe assay that basically what it does is it detects target molecules and it detects the relative abundance of those molecules with very high fidelity in solution. And I'll just go into detail on that. And I apologize if molecular stuff is not your thing, because you're about to hear a bunch of molecular stuff. So sandwich hybridization assay does not involve gene amplification or PCR. So it doesn't have the biases that are associated with PCR. It's also very, very sensitive, like PCR. It can detect RNA, DNA targets at very low levels. So what I mean by that is, for if you have a copepod, it's maybe 300 or 400 microns long, you're going to be able to detect probably one individual. If you have 100 micron across the nuclei, it might take 10, 15 to detect, but very low levels, which is great because we have tiny water samples, so not very many things sometimes. Probably tens to hundreds of copepods in one of those samples, depending. It's very robust. There's little to no influence from non-target genetic material. I'll show you some data that hopefully help convince you of that later. It quantifies the molecules, but not individuals, so you're counting them on RNA. That ends up being important later. And it's target specific, so it's not community characterization. You would not want to use this technique for that. Something like the high throughput sequencing, or the old school way of cloning method. And there are two formats for it, both the benchtop assay, which I'll be detailing, and the microarray version of that, which is only currently used on the ESP, but I don't see any reason why you couldn't use it on any microarray setup that you have. All of the ways to make all the reagents and do all of this stuff was recently published as a book chapter with Dave Carroll and Steven Stricker. This is available. I have this, so if anybody's interested in getting into this, I can definitely help you out. So let's look at the mechanics of how it works in a simplified format. This is the benchtop method. There's polystyrene solid support that's been bioaccumulated. The first of the two probes, the capture probe, is also bioaccumulated, and you add a streptavidin to that, right? So you get a 3-to-1 ratio of probes binding to anything that's coated with streptavidin, so the polystyrene counts. And then this goes into this plate processor thing. And when you look at this processor, if it's reminding you of the 70s, it should, because that's where it's from. And sometimes I think, I was born in the 70s, I love stuff from the 70s, so that's why this is in my life. And it's definitely got its ins and outs, but the company that makes it has just now come up with a brand new one. It looks a lot more like Robocop than this thing, so technology people will be happy. But the bottom line about it is the reason it's still around is it works great. So the mechanism continues for SHA when the capture probe hooked to the prong is introduced to a solution containing the RNA target molecules. Now, on those molecules, there's also a signal probe. That's our second probe, the healing site. And it's important for that site to be between maybe 75 and 300 base pairs distant from the capture probe. This kind of limits you in your probe design for a given gene. And we can talk more about that later. But basically, you're limited by the taxonomic variation that you have in a given gene, depending on what level of taxonomy you want to look at. So for something like the 18S RNA, this is not good for looking at, say, species level differences, but it's great for groups, great for the ordinal level of copepods. So the next part of the mechanism, you have a signal probe that it needles, and it's the oxygen label on both ends. And then the final steps, you have a horseradish peroxidase that's conjugated to an anti-DIG antibody, and that sticks to your signal probe and then mediates a substrate reaction. It gives you a photometrically quantifiable color result. And in the case of the ESP microwave version, it's not colorimetric, it's fluorescent. That's really the only difference. And this is what can be measured. And when we look at some of these dosage response curves in a little bit, I'll explain that in more detail. So what are we detecting with this right now in Bob's lab? We're detecting zooplankton. And we've got a couple of different examples of both holoplankton, things that spend all their lives as plankton, and meroplankton, things like these polychaete worms and crabs and echinoderms, bivalves, barnacles, various inverts that spend part of their life as adults on land or on the bottom, and copepods. And what we think we have here are very, very early larvation sacs. And so we now have a probe for detecting phagocytes and larvations, which is actually working out really well. Yeah.
Does that mean that you built or bought or borrowed the probes for each organism of interest? And now you have an arsenal?
We've actually, I've actually designed a number of the probes that we use currently, and also a couple of folks, actually also a fellow UCSC graduate, Joe Jones, who worked with Harvey for a while, developed about 3 that I use right now, and another former tech developed one as well. So yeah, we do have that arsenal. So I'll just make the point here that sandwich hive is broadly applicable. It's been used for all these toxic algae, Alison Haywood, Fisholin and friends, microbes, tabs. Chris Preston's been using it quite a bit for marine bacterioplankton, and Chris Sholin's lab. And some of you may know she was a technician at Bari for before that as well, doing this kind of work. And then Christy Smith in New Zealand is looking at invasives, North Pacific sea star, and done some zooplankton stuff as well. So you can use it for a wide range of things. The only thing that limits you is the taxonomic specificity that you can get for a given gene. I've been using these really fairly low specificity in the sense of variation, small subunit or large subunit RNA genes to do this, but we haven't had a real need for real high species specificity, except in the case of the green crab. Joe's managed to make one in the 18S that's, for all we know, at least for this area, specific to green crab. And these are the dosage response curves. So on the y-axis you have the optical density, OD, which we've seen as a measure of the RNA, essentially, because the DNA is there too, but it doesn't change very much. The RNA is the thing that varies and is most abundant. And you can see different numbers. 10 gets you up in the 0.5, 0.75 range here for this crab, and then for Millis, you need quite a bit more to get higher up. Smaller, maybe get more difficult, less RNA available. Polychaete, and also a balanced probe here that Freddie developed. So this was kind of my jumping off point, and I managed to kind of do the same thing for copepods. And so you see two lines here. The point of that is that we measure these things with a plate reader to get that colorimetric quantifiable data at the end of the run. And we do it 2 wavelengths so that we can have a wavelength that is a lot more sensitive and one that's less sensitive, so that if you have tons and tons of signal, you can move to a scale that allows you to actually see that variation. So the thing I'll point out about this is that I counted out numbers of individuals here, but I paid no attention whatsoever to the sex of the individual, whether it's gravid, if it's a female, or anything like that. And there's a surprisingly linear relationship despite that fact. So this owes the fidelity and consistency of this methodology. And as you'll see here next, we're going to get into some stuff that an intern a couple of years ago, Audrey Junaday, did. She was a fantastically hard worker, and it's some great stuff looking at the variation in physiology for Tigriochus californicus, and one of the things that I've noticed in the past, before she started this project, was that after a couple of years, the cultures give you, if you look at 1 versus a week versus a month versus 3 years, you notice that the early cultures give you a pretty even relationship between RNA signal and the numbers of individuals, but as the cultures get older, that starts to go down. This is something that Roman Marin and Chris Preston and Chris Schulman's lab have also noticed for pseudomycia cultures as well. To quote Roman, he thinks they're fat and happy, so they're just not physiologically not producing a lot, but the reality is that they could be suffering, or depressed, just not doing a whole lot. So for whatever reason though that happens, and we were interested to look into that. And so one of the things that Audrey did was she painstakingly weighed out individual copepods, and then we sent them down to a lab at UCSB to look at the total organic and inorganic carbon, and so that we could get a measure on the second y-axis here of micrograms of carbon for each one through some calculations, and you got your usual RNA on the y1, and then the x-axis is the number of individuals. And you can see for this 1 month culture, the first 2 lines, you have a nearly perfect overlap. Over the 3-year culture, the relationship between the carbon and how much RNA you're detecting changes dramatically. And you think of that as, per unit carbon here, you're getting the straight line, per unit carbon, you're getting a much lower response. And so that led us to do a few other experiments to see how we can tweak the physiology, or at least look at different kinds of physiology. And see, another way to look at this is more simply, it's just to put the RNA detection against the total carbon. And you can see, sure enough, that 3-year culture is a lot, the slope's a lot less than when you have a 1 month culture. So they're definitely producing less, even though all things size and all that are accounted for. The other thing we wanted to account for, though, was males, females, juveniles, and ophei in the case of copepods. And most of the females are gravid almost all the time, at least as far as I can tell. I'm sure that's not strictly true, but they usually have eggs with them, and so you have varying RNA amounts. In this case, very few females and quite a bit of RNA, and kind of even similar to males in some respects, but generally overall, females giving a stronger response to RNA. Total carbon's the staple there. And then the real interesting thing here is the nuclei. You can see barely detectable amounts of carbon for 20, 30 individuals, but the ratio of carbon to the RNA response is huge, because presumably they're growing quite fast. And we even counted out some fecal pellets because we were worried about signal being, available in the detritus and giving us different signals based on that. And we didn't find anything. The dotted line actually mentions the cutoff for what we consider legitimate detection. So I'll just show one more thing from this project, which is that what happens when you put non-genetic material in the mix. You have this great system, you say it looks great, but what if you start throwing other things at it? There's other stuff that things that are in this paper that I'm not going to show today, involving samples taken from the bay and mixed with things and then filtered and various things. But what we see here is a really high, 500,000 cells per liter concentration of pseudomycia, and by itself it doesn't breach this, it's similar to a control of filtered seawater here, but when it's mixed with the copepod here, you see the copepod on its own, and in the space with the copepod, you see a couple cases slight enhancement of the signal for the same number of copepod individuals, in other cases not. And the reason that we think this is going on is due to what I'll call a polyethylene glycol-like effect. And if you've done much about chemistry, especially in the past 20, 30 years or so, polyethylene glycol is something that's used to be added, maybe something that still is added to reactions. So you essentially boost the concentration of the target that you're trying to amplify or find. And what it does is it behaves similarly to large quantities of RNA and DNA, in that it has a way of organizing water molecules and kind of sequestering water molecules, effectively lowering the amount of water in a tube or in a sample that's available to the target, and thereby effectively increasing subject. It looks like that's happening. This happens also for the hab grows from time to time as well, but it's a small amount, so it's not something that we worry too much about. And I'll just basically throw in a plug here for a few ballast water samples. I'll thank Nick's lab for, Heather and Jules, thanks to them for providing these samples from the Cold Air facility. But this is just 1 liter samples compared, and I just happen to have some plates lying around, alkaline copods, kind of probes on there. And I was definitely able to detect both of those in the before treatment sample, and the after treatment sample that's comparable to negative control. So I haven't done much of that yet so far, but that was the first test, and it looks like it worked out. So I'm sure you guys have a lot of questions. We can talk about that more later. And so finally, I'd just like to give you some examples of some ecology studies the past few years. We'll talk about copepods and larvae in upwelling fronts, mussels and barnacles, and the nearshore conversion front, and then our current efforts to compare some of these novel AV methods, rheologic methods with more traditional methods. And so this is mostly the AUV stuff, as I mentioned before, and I'll start by some of the stuff I did when I first got to Mbari about 6-7 years ago, and basically this is a situation where we had no adaptive sampling yet, and we were just sending the vehicle out, telling it to sample at predetermined waypoints, and the idea was, let's head across the bay, we're sure to cross some kind of environmental gradient, and maybe we'll get something. And so what happened in this case, a couple of times we had your typical, Anyo and off tankers here, upwelling happening, and upwelling film extending down into the bay, and we kind of got lucky a few times and this transect crossed that. And so what I'll show you next is going to be the hydrographic data. This is a vertical view of the water column, and as you remember, the vehicle is going up and down, and it's sampling all this environmental data, temperature, nitrate, and chlorophyll at the same time that it's collecting these water samples here. And you can see on this map here where the shelf is during the first part of the transect, where you're out over the canyon, and then you're getting to be up over the shelf near Monterey on the end there. And one thing you notice as well is that the larvae, the mine signals for various larvae we're looking at, and copepods are shown as gray and black bars along the top, and these correspond to each of these samples taken. And so one thing you notice about the temperature, you can definitely tell where the upwelling film is, and the temperature gradient fronts. Remember, we had no adaptive sampling though, so we're kind of all over the place. But we did find that we found the fewest things out in the middle of that filament on average. We also found most things right along the frontal interfaces, in addition to chlorophyll, which is not too surprising. We have this intensity gradient with each nutrient supply, and we published these and other results I'm about to show you in a Gemini a couple years ago. Another way to look at these data is to do a principal components analysis on the AV samples, just using that environmental data, the sweep environmental sensor data into the vehicle. And so this part of this A panel here is that representation of that PCA. And what you want to do, you're probably all familiar with this to varying degrees, but visually the idea here is you have these variable vectors for the different types of environmental data involved, and you want to kind of superimpose this in your mind over this, such that the directionality of these vectors tells you near the top of the plot it's high salinity, near the bottom it's higher temperature, and those are inversely related, as they should be in this regime. And also high nitrate over on the right-hand side, and higher chlorophyll backscatter on the left, and more oxygen as well. And so what we found, showing some examples from what was in these samples and similar samples around them, over in the high nitrate upwelling column, we found very little that we had probes for. And then over on the other end, where the chlorophyll and backscatter are higher, although surprisingly also higher salinity, so this could maybe be seen as a mixing area here. The highest copepod signals we've ever detected. And then in the mid area here, probably stratified water column, moderate temperature, starting to increase chlorophyll. You have various copepods and more variety of inverts. They're all listed here, things I've pointed out before. So that was encouraging, and it led us to develop this upwelling front sampling protocol that I detailed before. And here's an example of a transect across the Northern Bay. And you can see that you do have this upwelling happening, and there's a front that it's crossing. And so in 2012, we got our first real big success with that. And so let's look at an example of how well Young's algorithm did this time. So what we've got on the top here are just the kind of OD on the left. This is the RNA signal again. And the organisms that we encountered sort of lined up with where the samples were taken. The upwelling core water samples are the squares over here, the front are diamonds, and the stratified circles once again. And this is chlorophyll on the top. You can see it's mixing across the front here. But in the temperature, you can really see where the temperature gradient is there. And basically, what we found were very, very low levels of copepods. And we also detected green crab in the front, which was, but it was interesting that the signals were so, we've never seen signals this low before. Looking at the remote sensing and weather, it turned out that the days preceding these studies was the strongest upwelling in about 40 years. And so all this work's done together with John Ryan, and so he's, doing a lot of this analysis with me, and we're basically concluding that there was enough water moving through the bay to really wash things out. So there just wasn't a whole lot in our samples. But nevertheless, when we looked at the samples from a perspective of things in the upwelling, things on the front, and then the stratified water, we did see that the front indeed acts as a sort of fence, a density fence, if you will, that there were some things that making it across there, and of course there are lots of organisms offshore as well that we don't have probes for, I'm sure, but that really the stratified water had the highest diversity and the highest intensity sample. So no huge surprises, but it's mapping with stuff that we already know from traditional studies. So at least we're not totally way off base, which is nice to know. It's worth knowing that.
Yeah, and this is based on a gulping device that samples about 1.5 liters.
This is only about 2 liters. You have 1.5 liters. So you're still getting. So as you'll see in a bit, when we're comparing these to net toes, it's really. I'm actually amazed that there's as much concordance as there is. It's, obviously things that are very, very abundant are going to be there in a small volume sample. So
is it one sample per site or station?
The way that the rod is operating right now is that it takes 10 samples in a transect, so you can put them wherever you want in the transect, but every time you're sampling just one. We do have some stuff that I'm processing right now where we've done all 10 at once, so that we can look at the non-sample variability to see what kind of things are going on. Another fun thing you can do here is you can arrange the samples by hierarchically by chlorophyll concentration, and of course the upwelling, upwelling samples have the least, and the stratified front has the most. You do see somewhat of a coincident apparent correlation between the count of copepods and that, which is not too surprising, but once again, it's sort of a double check. And so we talked a bit about copepods here. What about the larvae, invertebrate larvae? And we'll make the claim that fronts mediate larval retention, delivery, and recruitment, as I've mentioned before. And some examples from the literature include Roughgarden's work, who people are familiar with, and which included Mbari's own Tim Pennington, who, in my opinion, is a superstar. He works for Francisco Chavez now and does microalgae, but he got his start in zooplankton. So good on you, Tim. And crabs were transported along a thermal front. It's worked by Wing et al. And this is up in the Point Reyes area. Circulation near the front was a mechanism for retention of Brock Woodson and others. And then also from Brock's work in 2012, he showed that the recurrent ocean fronts, statistically speaking, set recruitment patterns for some economically important taxes. So we've been working also with Brock, and he's a co-PI on some of these projects as well. So I'll just give this example here. This is from early on, and this is the first use of the chlorophyll peak capture algorithm in this one. And we didn't really have a way for it to seek out fronts at this point, so we just set up this triangular AUV transect in the North Bay, and the hope was upwelling would occur, some kind of fronts would occur, and we'd see things happening near shore. And so what we did see, upwelling did happening, be working. And by October 2nd here, it moved enough inward and had enough effect to create a nearshore convergent front that this southeastern leg of the AUV transect, the triangular transect, happened to cross. This is thanks to John Ryan for many of these images. But what we also found, looking at the Maris maximum chlorophyll index and the near red reflectance, was that there was a red tide bloom that was kind of held near shore by this front. And you can see this drifter pattern here showing the recirculation that's going on. And yes, indeed, particularly around October 6th, this part of the transect, this southeastern leg, did indeed cross that front most directly. And so it's a chlorophyll peak sample now. And if you look at that same transect that is on the map, when we just split it apart and look at it for mussels and barnacles, and we have the calibration here, which may be a little bit fuzzy, but it's from the publications that I showed earlier, Joe and Sean's work. The highest signals are in bold, and you can see a lot of similarity between mussels and barnacles, which isn't too surprising. They've been documented to be fellow travelers in oceanographic studies in the past, but they're definitely occurring more along this transect and a bit along this one, not at all on the offshore, off the front angle. So pretty interesting stuff. And so what happens when we look at that more closely, try to get at the mechanism. And I'll give you a couple of examples here that John pulled out. In one case here, we have internal waves concentrating particles. And so if you look at the temperature plot in the lower left-hand corner, it's perhaps easiest to see these internal waves here. And you can see, looking at the backscattered chlorophyll, the concentration that's happening in this area here. And basically, the white line here is like a maximum kind of a backscatter isotyne. And then the gray circles indicate RNA signal for barnacles. And you can see that these black points have either very little or no, and that's just AV sample taken application without the biology. The largest ones were in these concentrated patches. And so another thing that can happen in addition to this kind of concentration, just incidentally, this is something that maybe you may have known for years because Jesus Pineda's seminal work on subject and people like Colin Shanks talking about internal waves and tidal bores, concentrating things and transporting them towards shore to be recruited. So that's sort of the next step in this scenario. Well, patch subduction and then shoreward transport. We also managed to detect and sample an in situ example of that. In this case, this is a 7-hour snapshot, so this is very, very high resolution. Tidal forcing, and we've got an incoming tide, and so you've got this subduction of this chlorophyll patch down to the bottom. The Dorado managed to sample it because it doesn't care about depth, it's just looking at chlorophyll peaks. No other method probably would have got that. And we did find the highest incidence of barnacles down in that patch, and then you see this tide sort of just pressing this shoreward. So it's, you got your temperature front here happening as well. So the bottom line is there's a lot of things that there have been tons of circumstantial and almost direct and really direct evidence for years. And we're in a position with these robots now to actually sample them directly and really kind of close the lid on that. So moving on, just this final section, I'll talk a bit about what we're doing now, comparing novel and traditional methods. Now, this is incredibly important, as several reviewers have made clear, that what you can do with a fancy pants expensive robot may or may not be worth anything if you can't relate it to some kind of reality that people are used to and that there's a huge body of work related to. So absolutely, we couldn't agree more. And along those lines, we've incorporated traditional methods into our sampling scheme. You can see that Dorado's sampling along this transect in the northern bay here inside and probably transcending the northern bay retention zone along the shadow functioning. And we've got a Niskin rosette sampling, a plankton pump coming off of that. We've got traditional nets as well. And I'll just mention that we've also called in some big known collaborators like Steve Morgan from UC Davis Bodega, whose work is primarily on crustacean larval transport. And we're also working with Bill Peterson. I believe you're on with Jennifer Fisher, who now works with Bill, used to work with Steve. And he's a copepod taxonomy expert as well. So we're in a position to use high-throughput sequencing on the nets and maybe even eventually on some of this stuff. We're in a position to compare these similarly volume Niskins, the little ones, the AUV gulpers, and we're doing things like we've got a transect here where the AUV's targeting chlorophyll and just hitting the layer all the way along. And then we're also following that same line with the boat with CTD casts and using a human in the loop to sample above, within, and below the chlorophyll layer at each point. So we're really trying to hit it with everything we know how to and get some real good comparisons going. And so an example of that data, how did the Niskin and AEV data compare? So you see a variety of things over here, and you can see the points where the Niskins are being sampled and the CTD path and the AEV transect. We actually have two AD transects, one down here as well, other Niskin stations, but we haven't done that yet, so it's not on here. So for the Doppler samples, you can see that this is daily variation roughly at each of these these locations. Those vary a little bit, and Niskin is below. And Niskin is a little bit more intensity, which I think can be owed to the fact that these are a couple of 2.5 liters versus 1.5 liters, maybe not sure yet. But similar patterns are emerging. Things like sand tube worms and barnacles are occurring kind of more frequently and kind of out a bit, and also near shore. Copepods, of course, are everywhere. Don't be surprised by that. And also brachyurin crabs occurring in one case here. Echinoid and tunicate larvation probes, the new probes we have are kind of dominating the samples. So that's pretty interesting. We're looking into that right now. And then an example also from what the net toes look like around the same time. The same stations where the Niskins were taking all these net toes. And so what you've got here are the same kind of thing, daily variation over a 4 or 5 day period. And barnacles also more near shore, these sites, copepods everywhere. And then interestingly, I don't have a probe for them yet, but euphausids taking place here, and then once you get out past the retention zone, not so much. And then also this is diatomaceous, includes things like pseudonychia in the mix. So we just got this data in. This is 454 piracy data from Korea, and we've got something like, I think it's 68,000 bidirectional reads. That's after we clean it up and throw away tens of thousands of other reads. So it's a lot of data, and Shannon Johnson's working on that right now, bringing that all together. So we've made some errors, and we got it for 28S, but also doing it for CO1 right now too. So it should be pretty. Yeah.
Do you mean copepods in bulk altogether, or for the suite of tags that you have for the species that you're considering? I'm confused with that.
Oh, okay. So let's see if we can find the source of that confusion. So do you mean you're wondering about what was in these samples?
Well, the word says copepods.
Yeah.
Have you now?
Well, what copepods are these?
Is that the blue that you happen to have tags for?
So here's the deal there. This blue actually represents all of that stuff that I have tags for, and a lot of other stuff as well. Probably the stuff that I have may represent, I'm just guessing here, 20% or 10% of the stuff that's shown in blue here. Although, I don't know that. And these are, I believe when she did this, because of this picture here, I'm pretty sure I remember our conversation, these actually are calmer copepods specifically, which is winnowing down quite a bit from the greater, community. There's a lot more stuff there. So this massive data, and I know John tells some of this as well, it's a bear. I mean, we just barely figuring out how to do it. We're using a program, Shine, that seems to be working out pretty well. A lot of the software is readily available. There's a bit of a software engineering involved, but we have people for that, so we're taking advantage of it. And wanted to finish up by saying that we've got these unprecedented spatial and temporal scales we're able to sample now. We've got a robust method, among other methods, that's well suited to a variety of applications potentially. And actually, I was just up at NOAA yesterday giving a talk because there's some river ecologists out there that want to maybe use this technique to look at caddisfly larvae. One of the things about sandwich hide is that if the water type doesn't matter. I even think you could get things out of soil samples and do it. It's really robust. It doesn't seem to care. There's no enzyme mechanics involved, so the conditions can be pretty rough and it still works. Comparison to traditional methods is important, links to previous work, and these methods are contributing. So with that, I'll thank my funding sources, and I'll thank all of you for giving me the time to tell you about this crazy stuff. I'm happy to discuss it with you, probably at a length greater than we even want to.
So Chris developed the sandwich hybridization with ribosomal RNA as target. Have you used it on other things, single copy genes or CO1 protein coding genes?
That's an excellent question. And I actually had a comment yesterday that maybe you could use it for analysis, you know, if you had to, because it's very, I mentioned earlier, I didn't mention it today, but a single base pair mismatch kills it for you. It won't stick, and you're good. But yeah, we have looked at that a bit. Before my time, Joe Jones tried to make some probes for CO1. You can imagine he wants that. It's no stretch of the imagination. So he tried to do that. He never got very far with it. I think it's because he switched jobs, possibly. But also the nice thing about ribosomal genes is they're really high copy number, right? So the chances of getting something greater. I think it could still work on other groups. And to be clear, Chris's crew also, they use 28S a lot, which is a bit more variant than large sodium, as you know. But by far and large, people stuck with those. And I don't know if it's just because, hey, they're answering the questions we need to answer, and we're not going to forge ahead because of that. But the future is out there, so it's possible.
I always thought it was because of the high copy nature,
Yeah.
but it's not the ideal target for species level or population level characterization.
That's absolutely right, and that actually brings up something I don't know if I emphasized enough. I tried to, but this methodology is really good at doing a couple of things. It's really nice not to have that application bias because you're looking at real amounts of what's there in the solution in terms of what we've sampled, and it's also great looking for what you want to look for. But things like really high species specificity might not be the best thing. Definitely not good for community analysis. Unless you only care about a few things, like these river, Eric Danner and some folks up at NOAA, they have a model they're trying to inform, and they have like 3 or 4 groups, like carnivores and herbivores and things like that. And in each of those groups, there's only like 1 or 2 species that they really care about. And I guess it's pretty depopulated in some of these environments. So in a case like that, if you want to do thousands of samples, you want them all to be really consistent, and you don't have the money or the time to pay technicians to sift through, thousands and thousands of items from a plankton net that frequently, you can take a few plankton nets as a way to ground truth this, and then you can do this with a lot larger number of samples. So I would actually say that for something like this, you really have to have that, morphological sample as well to kind of give it validation, regardless of what you're working on. Yes.
And I have naive questions on
My favorite kind.
but I couldn't help but notice the y-axes had tremendously large optical densities, like 1,
Yeah.
3, 7. That's like black.
Yeah, so here's the deal with optical densities. There is a limit to this thing. And one of the problems that you're going to run into if you try to use it on larger organisms or large volumes of water with lots of organisms is that there's a max limit to it. And when you get up around 3 or so, the scale goes up to 4, and the machine stops being able to differentiate it for you. It really is black at 4.
Is the color, you mentioned the word
Yeah.
horseradish, is the color basically produced in the same way you would produce it in a ELISA?
Yeah, it is, and that's the technology we're talking about here. Yeah, it's all, and that's, I guess there's enough people still doing this sort of thing to where, chemistry stuff, to where these reagents are still available. But the specialized reagents for this, I should mention, are something that we have the ability to make in-house in Mari, and that I've published this chapter to allow anyone to make, and you can do it, with the exception of one of the reagents that are all fairly inexpensive. But the problems are that there's a company that actually Cygen makes the machines that actually has in the past provided all of the reagents, but their availability has waxed and waned. And there was last year they changed hands or something, and they weren't making for a while. And so we went back to making, and actually recently I just went and made my own stuff. And depending on your setup, that might be more cost effective, but you can do both.
And this is connected, this will help me understand the best then. So, and I think I have it, but I just want to make sure. So in a sample, when you gulped and sipped it, if on the filter that you're going to do the extraction out of, there's one Calamus pacificus nuclear stage 2, and one C5 copepodite, 1,000 times larger.
Yeah.
Who wins in the signal producing?
If you're, are those the same, are those different species?
No. So I just want to know is this biomass specific response, like
It's
one
gonna be
nuclear
that.
stage
Yeah.
of these species and one copepodite stage of the near adult?
Yeah.
Well, one's got a
The
thousand
big
times
ones,
bigger
the
than
big
the
one
other.
is where you're gonna get your signal. Let's put it this way, if you separate those onto two filters and do two assays, you're not gonna detect a single.
You won't even know you hit the
You won't know
nucleus.
it's there.
Yes.
It's just one guy. And on the other guy, you're gonna get bare level detection probably if your probe moves.
But you've also said that it's, so if you had a collection of nuclei, you would detect it, but it's not a linear thing because the younger ones are making more RNA.
Yeah.
So you need some sort of calibration there.
Yes, because it was very interesting to
Yeah.
see copepods stated as a group.
Yeah.
But there's a couple of caveats
Yeah.
inside there because the individuals are producing the signals,
That's
and
right.
not only the individuals in that plot, it was the species
Yeah.
specific production of the signal as well.
Yeah.
It was not collectively looking for something generic that I don't know about, like
Yeah.
a generic copepod tag.
Yeah.
Wrong, correct?
Well, some of the copod probes that I have that I display here for the sandwich type at least are they're very broad taxonomically. What I did was I actually, in some of the cases, I divided up what I could get from the database, which was substantial, the Caloida, into different sections. And how did I do this? It was not totally arbitrary, but what it was based on was the variation in gene I was looking at. I found that due to the other constraints, I could make a probe over here that got these orders. And I could make a probe over here that happened to get these other orders. And I could make a probe over here that just got this one order, that sort of thing. So you're limited in that sense by what you're looking at, the available data. And the real tried and true method is if there's something that you want to detect real specifically, or you want it to at least be included in the things you're detecting, that you want to, what you need to do is you need to just grab that thing and quickly sequence the gene you're going to use so that you absolutely know the sequence for the thing that you want to detect. But John's point is a really good one, and that's that when you have a scenario where let's say you have a really high signal over here and a medium signal over here, you don't know if it's copepods, at least especially the ones that we did the work on. You don't know if that's 300 nuclei or like 10 adults, or whatever it is, or I'm sorry, invert that, if that's 20 nuclei or 40 adults, So that's going on, and if you need to know the demography like that, then you're going to need to look at it. Yeah.
I was just wondering, would it be possible to elute your RNA off of your probe and then do the traditional clone and sequence and see how taxonomically specific your probe really is?
You can totally validate it. There's been work where we've taken, the one thing I didn't mention about this method that's kind of nice is you can take the lysate after the plate runs. It's in like anywhere from 1.5 molar to 3 molar of monodynium. So it's pretty well preserved. I mean, it's like the carbon freezing of lysis buffers, right? And so after it's been on the bench for now, it's like even being heated while the plate's running. But you can take it, you can freeze it, you can do an isolation from it, you can get out DNA and RNA, you can have a fine sequence.
But the lysate is what's left behind after the probe has removed your target. So you get your target off the probe.
There's enough, generally, if you had really low levels, you might not get anything, but and you're right, it would depopulate that, but there's a limit, I think, to how much is sticking to the prongs, and in cases where you've got a strong signal, there's definitely some left over, I think, in some cases, potentially, just because.
If you sequence the lysate, you don't know what you had on the probe, so you can't compare.
Oh, well.
You can't validate the probe
Here's
with the
thing,
lysate.
like, if you think about I think the thing that's where I'm troubled here is that the probe, the prong surface, or if it's a microarray, the surface of the amount of molecules, the finite number of probe molecules hanging off of that thing, when that's saturated, it stops taking. So if you're above that limit, you actually are going to leave behind target material in the solution.
Right, but I guess what I'm getting at is the lysate has the leftover of your target, but it has the leftover of everything else too.
Yeah, that's right. You're right. So I guess the idea would be like if you're saying, for example, okay, I wonder if they're really got green crab. So then you go ahead and you sequence stuff from the lysate. If green crab's in there, you think, okay, great, but it's actually not answering the question that you're talking about.
Well, yeah, so you show that there's green
Yeah.
crab in the lysate, but if you
Yeah.
also found cancer and,
Yeah.
Pugetia,
Yeah.
do you know that your probe
The
did not?
Yeah, the only way to do that, and that is something that is worth doing in a situation where you're differentiating among the other ones, and then you'll actually do that with this probes. You have to get samples of those adults. The best way to do is get samples of the adults, generate some RNA in solution, and then do like comparative testing, and just figure out, you have to show that.
Yeah.
So in situations, for example, in situations where the thing that you're after is kind of off there in a vacuum by itself taxonomically, it's no worries. Not because the probe is really that specific, but there's nothing else there that's going to pick up anyway, so you're fine. But in a lot of cases, like crabs, and there's a bunch together, you would need to, the best thing to do is to take an ETO maybe. Not that you can identify crabs of that size, but like I said, adults would probably be the way to go. And you say, okay, I know all these are extant in this area. I'm going to grind some up from each one. I'm going to do side-by-side tests till I convince myself this thing isn't picking up the other ones. And Sean did that with barnacles as well. She picked 4 or 5 common species. And, you do the one that's not the target. In some cases with hers, actually, she found that I think it was thalamus and maybe one other one. She basically wrote on thalamus. And it actually did cause a little bit of reactivity, but she maxed it out, and it never gave a signal above like 0.15, 0.2. So she just made her cutoff higher for detection. And thalamus did a nice curve all the way up. So.
Come on, somebody ask a question.
Cool.
I have one. So would you by chance know how fast RNA deteriorates or goes away after an organism is killed? Thank you, by the way, for the ballast data. Does it make a difference if you kill it with like UV or chlorine, and would it take the RNA out faster, or after senescence, like how long does RNA stick around for?
Right. RNA does not stick around very long at all. It is, in my, just from the literature and from my own experience, it degrades very quickly. An example of that is I was briefly involved in some microbial studies on SLU a number of years ago. And the deal was you take a mud sample, liquid nitrogen flash freeze it, put it in the -80, or keep it in liquid nitrogen until you use it. You have one chance to thaw that thing out and use it for RNA, and it was done. You couldn't use it for RNA after that. For bacteria, okay, which are a lot less complicated, but the same thing goes for eukaryotes. And the bottom line about this sampling and about these methods is that you are kind of limited in that you need to get those samples on a filter. And if you've got a large organism that's not going to die right away, maybe it's got 15, 20 minutes, as long as it's alive, you're fine.
Mm-hmm.
Unless you're trying to, work on some kind of stress-related gene, then I think you'd be in big trouble. But the bottom line is that once the cells die, RNases are everywhere. It's a done deal. So it does need to go right into RNA later or liquid nitrogen, I think, as well.
So it's safe to say then that there's not much free-floating RNA out in the ocean, or?
That's actually a good question. I'd be hesitant to make any general claim about the ocean, but the thing about free-floating RNA in the ocean, there's a hell of a lot of viruses in the ocean. So I don't know what that does for you, but basically, I actually know some folks that work on those, and that's a different scene. But in terms of like these macroorganisms, we've been real worried about environmental effects, and some of the stuff that came out of that 2013 paper with the intern Audrey, we did some tests where we did the same dosage response curve against background water that we collected, both off of rate 4 and N0 and N1, and looked at how it caused that to fluctuate. And then we filtered those same water samples to like 100 microns to take out obvious naturally occurring targets, and brought the curve right back down in line pretty much with the non-environmental curve. So, there's a lot of, the probe specificity is an issue, but the tests we've done with seawater, filtering seawater at large porosities that definitely allow any genetic material through, there's never been any problems with that. And I think for eukaryotes especially, RNA is not going to survive. It just gets chewed right up.
Cool. Thank you.
Well, we'll thank Julio, and stick around
For a minute or two.